Hello friends, this is the fifth class on genetics, and in this class I'll be discussing on the DNA replication. Now we have to know about the central dogma, that is when DNA is replicating itself, then it is called the replication. When DNA is producing RNA, then this process is called transcription. Transcription. I'm really bad with my handwriting. So this is transcription. And from RNA, when you make protein, this mechanism is called translation. Alright, I think you can understand what I want to write. It is replication, this is transcription, and this is translation, and whole this phenomenon is together called the central dogma. Okay, so this is the thing that is responsible for production of new DNA. Then from DNA to RNA, this RNA, three types of RNA are there: mRNA, rRNA, and tRNA. And then from with the help of these three RNAs, this mRNA encodes protein. Alright, so now we will be discussing the part of this replication both in eukaryotes and in prokaryotes. Okay, so now let's go to that portion and let me clean this up. Okay, alright, so now here's the replication, and we will be discussing this replication in prokaryotes and in the eukaryotes as well. So I'll be starting with the prokaryotes. Now, thing is that uh, suppose this is a DNA strand, double strand DNA. This is the 5 prime end, this is the 3 prime end, and this is again the 5 prime, and this is the it is the 3 prime, and this is the 5 prime, because this is anti-parallel. So uh, in a DNA, before replication starts, one has to understand that there should be a specific place from which the replication starts, and which is called origin of replication. Okay, this term is important because this origin of replication is highly a restricted sequence, and it controls the replication process very uh, sensitively. So a unit of DNA in which replication starts from an origin and proceeds bidirectionally or unidirectionally is called a replicon. Now if this is the DNA strand, and suppose this is the place where the replication origin, this is the sequence, is a start point, and uh, the DNA double helix, the bonding between the the two strand is the hydrogen bonding. Where the replication will start, the hydrogen bond will melt, and one bubble will be formed, which is called a replication bubble. All right, and this bubble can can move in this direction or can move in this direction. So this and this portion, this look like a fork, because if you take the half of its view, like this portion, if I look at this portion, it is just like this, and if you take a look in at this portion, it's just like this. Okay, it's like this. All right, so in that case, this portion is not there, sorry, this portion is not there, this is not there, so it is just a kind of a straight line, and it looks like a fork. So this is the fork which goes into this side, and this is another fork that is going into this side. So this is a bidirectional fork, or sometimes, if the fork is moving only in one direction, suppose it is not moving in this direction, only this direction is the fork is moving, so this is called a this is a unidirectional fork. So in that case, this whole unit, this I'm talking about this whole unit, where this replication origin, I mean this DNA melting and replication bubble takes place, and the fork is formed. This whole section or whole segment of the DNA is called the replicon. Okay, so this replicon uh, in prokaryotes is usually circular. Okay, now the thing is that because the DNA in the prokaryotes, if we take an example, the bacterial DNA, which is usually circular in structure. So this is the circular DNA. No, I should take a structure and should draw this one. Suppose this is the structure. I'm not really good at it. So if this is my DNA, this is my first outer strand, is my inner strand. So suppose if this is the structure, this can be the replication origin, or we can say that we can call it as a Ori C. Now, this Ori C has got specific sequences which makes them different from the rest of the sequences, and which can be targeted as the start site of the replication. So I would like to go into that section of our discussion, and this region where the replication starts is called Ori C, origin. Okay, so this section, this the segment of this DNA contains repetitive sequences. If I want to draw this one, it looks like this. That suppose if this is my DNA and this is the 5 prime end, this is 3 prime, this is again 3 prime, it is 5 prime, then there are 9 base pair containing repeat sequence, and there is another 13 base pair containing repeat sequence. Now this is my 13 base pair containing repeat sequences, and there are 3 sequences like this which has got 13 base pair, and those 13 base pairs are like this. This is G A T C T A T T T A T T T. So now if I this is 4, and this is again 4 8, and this is 4 12, and this is 13. So this is the 13 base pair sequence, which is a repeat sequence, because there are, you know, three copies, 1 2 3 copies, which contains this specific kind of sequences, and another one is a 9 base pair long sequence. This is the sequence which is a 9 base pair long, and 4 sequences are like that, and the sequence is, you know, this T T A T C C A C A. All right, so this is my 13 base pair long repeat sequence, which is three there are three segments which has got this repeat sequence. The sequence is, that's why we call it a repeat sequence, and 9 base pair containing repeat sequence is there, and this is the sequence. All right, so uh, this one is called a 9 Mars, and this one are called 13 Mars. Now now the thing is that these sequences are highly specialized in binding some specific protein which are necessary for this DNA replication. And like, you know, the first protein which is very important is the DNA A protein, which works as a helicase. Now what is helicase? I would like to talk about that as well. The helicase is, as we know that this DNA structure, this is the DNA helix, and how this helix are bonded together. If this is the 5 prime, this is the 3 prime, and again this is the 3 prime, and this is the 5 prime. I believe this is the strand that I have drawn is fine. No, it's not 5, it should be 5 prime and this should be 3 prime. Anyway, so this is a strand of double helix DNA, and this each strands are bonded together by this hydrogen bond in themselves, and which actually stabilizes this double strand structure. And when we have to do this kind of a replication, we have to unzip this structure. So for that reason, this hydrogen bonds in between them, if this is not a helix structure, this is just a ladder kind of a structure of the double stranded DNA. And what happens, if we have to make a replication bubble like this, that means in that place we have to break the hydrogen bond. We have to break the hydrogen bond, or and as the replication fork will move in this direction, that direction, again these hydrogen bonds have to be removed or have to be broken. So this kind of phenomenon, which is called a DNA melting, is done by the enzyme called helicase. So here is the enzyme helicase, which actually functions to melt the DNA, to break the hydrogen bond in between them. And this DNA A is the enzyme which works as the helicase, and this is very important. Now this DNA A protein, it actually binds to this replicon, to this start site of the replication. And this is the place where the DNA first unwinds. Now there's one very interesting or very informative very important information I should be giving you, that when this DNA replication can start. Suppose if my as we have already known that this DNA is a semi-conservative replication, that means if this is my DNA strand, which is the original DNA strand, and if suppose I'm taking a new color, okay, this red is fine. So this will be my one daughter strand, and this will be the parent strand. So so now after the replication, two new molecule will be originated from the original molecule, and one of the strand will be the new strand, the other strand will be the old strand, parent strand, we can say. And here this is also, the one is parent strand, this yellow one, and the red one is the daughter strand. So this kind of replication is called a semi-conservative replication. If it was a conservative replication, then the things would have been different. Then from our single parent molecule, we would have got one molecule that is completely made up of the parent molecule, parent DNA, and the other one would have been completely made up of new DNA, that is daughter DNA. But this doesn't happens as if we know the Michelson-Stahl experiment. We can discuss it in a different video, but here I will only say that the Michelson-Stahl experiment showed that the DNA replication is a semi-conservative replication, and this is the example of the semi-conservative replication, and this is the example if the DNA replication would have been conservative. So now again, the thing is that what I was trying to tell you, that how we can how the DNA can be identified. What is the exact time when I will start my DNA replication? Okay, this start sequence is highly specific, and it is restricted in case of DNA replication, as this section, this GATC, this section is, if it remains methylated in the oldest strand, and when it is methylated, then only the DNA replication can take place. And if it is not, then the DNA replication cannot start. I'm lacking time, so I will see you in the next video, and I'm quitting now. Thank you.